In this video, we will discuss about the experiments where the researchers has analyzed whether FAS receptors, that is the dead cell receptors, plays a critical role in the eradication of cancer cells, and why they come up with these experiments. Because in the previous video, we have understand that KRAS is inversely proportional to the FAS receptors, and when the KRAS goes down, they have observed the eradication of the pancreatic cancer cells. So from that experiment they come to a hypothesis that it's the FAS receptors, that upregulation of the FAS receptors on the cancer cells with a decrease in the with the elimination of the KRAS results in eradication of the cancer cells, as this FAS might interact with the FAS ligand on the immune cells, and then that will trigger apoptosis of the tumor cells. In order to check that hypothesis, they did some experiments where they inhibit these FAS receptors and check whether it reverses this eradication of the pancreatic cancer cells or not in the KRAS eliminated. So let us start on the results that they are presented in the paper. So in the panel A, we can see that they just validate that their design is working as per expectation, and they use 5 different kind of short hairpin FAS RNAs to suppress these FAS receptors, and those results are shown in this panel A, where we can see that the KRAS mutations are on. I do not see any difference in the FAS receptors, neither in the parental case where no injection of the FAS receptors is there, and neither in the case where FAS inhibition of the FAS receptors using short hairpin FAS receptors RNAs are used. And they, for control, they used the scrambled short hairpin RNAs. But when the KRAS mutations are off after 3 days, they see an increase in the FAS receptors in the parental case, and also in the scrambled short hairpin RNAs, but not in the scrambled FAS receptors. And this increase in FAS receptors further goes up after 5 days of the elimination of the KRAS, and we can see that there is a further increase in the FAS receptor signals in the parental case and the scrambled short hairpin RNAs, whereas the FAS receptors is statistically lower in the case where short hairpin RNAs, again short hairpin FAS receptors RNAs are used. And these things, they are also validated using the Western blot, where we can see that the KRAS mutations are on, they do not see any FAS receptor signals, but when the KRAS mutations are off, they see the FAS receptor signals in the parental case and the scrambled RNA, but not in the case of short hairpin FAS receptors RNAs. And even after 5 days, the signals of the parental and shRNAs FAS receptors goes up, whereas there is no increase in the signal of the FAS receptors in this SH short hairpin FAS receptors RNAs cell lines. And after validating that their design is working as per expectations, they wanted to do their experiment in vivo in the mouse model, that is C57BJ or 6J mice models. So they did 2 different kind of experiment. In one case, they continue to inject doxycycline so that KRAS mutations are on throughout the experiment. In the other case, they inject KRAS mutations they inject doxycycline to have KRAS mutations for 10 days, and after that the KRAS mutations are off for 20 days. And in this case, the pancreatic cancer cells are introduced using orthopedically. And what they have find out is that in the case where scrambled RNAs are used, or scrambled RNAs on and off are used, or we can say that is short hairpin FAS receptors are used with the KRAS mutations are on, and the short hairpin FAS receptors RNAs are used when the KRAS mutations are on and off. We can see the MRI results, where we can see that in the parental case where scrambled RNAs are there, after 10 days there is some tumors volume, and this volumes is also seen in the case where scrambled RNAs are there after 10 days, and when the FAS receptors are there, they see the tumor volume is high. The tumor volume is shown in this dark white color. So tumor volumes compared to the case the parental or scrambled RNAs is higher in the case where KRAS mutations are on and FAS receptors are goes down, and when the FAS receptor and the tumor volumes is also visible in the case where FRNAs are there and KRAS mutations are on and off. This panel top row is about the 10 days, and after day 20 it's about the after day 20 they show the similar mRNA results, and here we can see that change in the tumor volume. In the case where scrambled RNAs are used and the KRAS mutations are on, we see that tumor continues to rise. When the KRAS mutations are on and the scrambled mutations are there, we see the tumor volumes goes down as the FAS receptors might have goes up, and in that case, since the KRAS mutations are on, the tumor volumes continue to increase. And whereas in the case where FAS receptors are there, KRAS mutations are on, they see that further increase in the tumor volume. Whereas in the case these FAS receptors are KRAS mutations are on and off, whereas FAS short hairpin RNAs against the FAS short hairpin FAS RNAs are there, they see the tumor volumes are increased. So don't focus on this dark white color, but focus on the region that is shaded by the yellow boundary. This indicates the tumor volume. So we can see that tumor volume goes down when the KRAS mutations are off and scrambled short hairpin RNAs are used, whereas when the tumor volumes goes up in the case where KRAS mutations are either on for a whole experiment or KRAS mutations are on and off where the short hairpin FAS receptors are there. As the FAS receptors are not available, the apoptosis triggered by immune cells does not occur, and these things are quantified in the panel E, where we can see that the tumor volume, there is no significant change in the tumor volume when the KRAS mutations are on or the KRAS mutations are on and off, or we can see that the scrambled short hairpin RNAs are used with the KRAS mutations are on. So in that three cases, there is no change in the tumor, significant change in the tumor volume. This indicates that because the FAS receptors values or FAS receptors on the tumor cells does not change significantly in this case. Whereas when the scrambled RNAs are used and the KRAS mutations are off, we can see that tumor volumes in this case also does not change significantly, because there is no factor to increase the there is no factor to decrease the KRAS mutations. So in all three cases, in all these first four cases, as soon as the all these first four, let me re-explain this panel. In the first case, when the KRAS mutations are on, we do not see any significant. There is a control case. So these are the amount of tumor volumes that we have observed. But when the KRAS mutations are off, we see a slightly decrease, but this decrease is not statistically significant, as because with the decrease in the KRAS eliminations, we see that there is a when the KRAS mutations are off, there is an increase in the FAS receptors, and that leads to a decrease in the tumor volume. And when in the case of scrambled RNAs and when the KRAS mutations are on, we do not see any increase in the FAS receptors. Whereas in the panel B, as soon as the whereas in the results B, as soon as the KRAS mutations goes off, there is an increase in the FAS receptors. Whereas in the case of in the third case, there is no increase in the FAS receptors and the KRAS mutations continues to on. Whereas in the fourth case, when the KRAS mutations are off and the scrambled short hairpin RNAs are used, so as soon as the KRAS is off or the KRAS mutations are off, we see the increase in the FAS receptors that will cause tumor death or apoptosis of the tumor and leads to the decrease in the tumor volume. And in the case where short hairpin FAS receptors are used and the KRAS mutations are on, we see the continuous increase in the tumor volume, or percentage change in the tumor volume increases, because the KRAS mutations suppress the FAS receptors, and the short hairpin FAS receptors mRNA also suppress the FAS receptors. Whereas in the last case, where the SH short hairpin FAS receptors continues to suppress the FAS receptors, the KRAS mutations are off. So in that case, only one factor plays a role. That's why the change in volume is slightly lower than the second last case, but it is not statistically significant. The lower is not statistically significant. So it indicates that this FAS receptors plays an important role towards this eradication of the pancreatic cancer cells. And these things they are validated using the immune cells infiltration, that is T cells infiltration or CD4 plus cells infiltration. And they find out is that the KRAS mutations are on the amount of CD4 plus cells in the parental scrambled RNAs and FAS short hairpin FAS mRNAs are lower, but when the KRAS mutations are off after 5 days, we see the infiltration of the CD4 plus and CD8 plus T cells in the parental and short hairpin scrambled RNAs, but not in the case of the short hairpin FAS mRNAs case. Indicates that FAS receptors are not there, so immune cells will not be able to perform its function, or immune system will not act similarly as it acts in the parental case and short hairpin scrambled RNAs case. And these things are quantified in the panel I, where we can see that CD4 plus cells goes up after the KRAS mutations are off in the parental case and the scrambled RNAs case, but not in the short hairpin FAS receptors case. And similar results they have seen for the CD8 plus T cells also. When that is CD8 plus T cells goes up, the KRAS mutations are off after 5 days in the parental case in the scrambled RNAs case, but not in the scrambled FAS receptor case. All this indicates that the FAS receptors plays a critical role, as the immune system also has this FAS ligand that can interact with these FAS receptors on the immune cells and trigger apoptosis. So I hope you understood how they validate their hypothesis that FAS receptors plays a critical role in eradication of cancer cells. If you need to understand more about their experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.